So in previous class, we were discussing about the viruses, right? So in today's class, we can discuss about bacteria. Okay. So listen, prokaryotes, they thrive almost everywhere. They are including places to acidophilic organisms are there. They are loving salty. They are loving cold condition or hot condition, thermophilic organism, kynophilic organism. So likewise, the prokaryotes, they can live. Prokaryotes are omnipresent. We will tell that particular term, when an organism is present in any habitat, in anywhere, if you can see an organism anywhere, you can call such organism as omnipresent organism. That is omnipresent. Omnipresent. We will call it as omnipresent. Omnipresent organism. Such organisms can be called as omnipresent organism. So an organism, the omnipresent organism, such organisms will be present everywhere. You can see this type of organism everywhere. Such organisms can be called as omnipresent organism. We will call it as omnipresent. Okay. Look into the next point. There are more prokaryotes in a handful of fertile soil than the number of people who have ever lived. So if you are looking into the number of people started from the origin of life till now, more than that number of organisms can find in a handful of fertile soil. They have an astonishing genetic diversity, different type of prokaryotes that you can observe. The prokaryotes are divided into two domains. We can classify them as bacteria and archaea. We can classify them into bacteria and also into archaea. Bacteria are there and archaea are there. These are the two types of prokaryotes. Two types of prokaryotes are bacteria and archaea. Okay, clear? Ready? Moving into the next slide. So, most prokaryotes are unicellular. Almost all the prokaryotes are unicellular, although some species form colonies. The microbes, they are having the ability, the prokaryotes are having ability. When they are grown in a nutrient medium, when they are getting a nutrient medium, they will grow, multiply, and they will form colonies. Most prokaryotic cells are 0.5 to 5 millimeters, much smaller than the 10 to 100 millimeters of many eukaryotic cells. What is the difference between eukaryote and prokaryote? That is very clear. A prokaryote will not be having a well-defined nucleus. Nucleus will not be present in the case of prokaryote, but genetic material will be present. Whereas in the case of eukaryote, the genetic material will be present inside the nucleus. Prokaryotic cells, three most common shapes are spheres. Coccus organisms are there, bacilli, that is rods, and spiral organisms are there. So on the basis of their shapes, we can classify them into cocci. You might be heard about staphylococci bacteria, which is having spear-like appearance. That is cocci bacteria. And bacilli is the Bacillus thuringiensis. You might be heard about another organism. Anyhow, bacilli organisms are there and spiral organisms are also there. If you are looking into a spherical organism, this is a spherical organism having the size of 1 millimeter, and rod-shaped bacteria or prokaryotes having 2 millimeters, and spiral, which is having 5 millimeters in their diameter. An important feature of nearly all prokaryotic cell is their cell wall, which maintains cell shapes, provides physical protection, and prevents the cell from bursting in a hypotonic environment. So it is one of the most important feature of a prokaryotic organism that they are having a particular cell wall by protecting that organism's body. In a prokaryotic organisms, almost all the prokaryotes, we have told that even though they are forming colonies, prokaryotes are having a single cell only. So all the metabolism should be happen inside a single cell. For example, if you are comparing a human being's body and a bacteria's body, all the things that should be happening in a human being's body, such as reproduction, such as digestion, in that manner, several chemical reactions are occurring in our body, right? All those reactions have to be conducted by a single cell only. So in order to protect that single cell, they are having what? They are having cell wall, which maintains cell shape and also provides physical protection and prevents the cell from bursting in a hypotonic environment. Whenever it is present in a hypotonic environment, there can be the chance of entry of water molecule into the cell if there is no cell wall is present. So as it is a cell wall is present, there water molecules will not enter into the cell and it will not cause the cell to burst. Eukaryotic cell wall are made up of cellulose or chitin. It is made up of cellulose or chitin. Both are polysaccharides, we know. Bacterial cell wall contains peptidoglycan. Bacterial cell wall, if you are looking into bacterial cell wall, that will be containing peptidoglycan. Peptidoglycan, from the term you can understand it is containing amino acids and also glucose molecule. A network of sugar polymers cross-linked by polypeptides. So protein molecules are present there and also sugar polymers are present there. That is a cell wall of a bacteria. Using Gram stain, scientists classify many bacterial species into gram-positive and gram-negative groups based on their cell wall composition. So based on their cell wall composition, we can classify the bacteria into gram-positive bacteria and gram-negative bacteria. What is the difference? Gram-negative bacteria have less peptidoglycan and an outer membrane that can be toxic, and they are more likely to be antibiotic resistant. In the case of gram-negative bacteria, they will be having less peptidoglycan and an outer membrane that can be toxic, and they are more likely to be antibiotic resistant. So gram-negative bacteria are antibiotic resistant bacteria. Many antibiotics target peptidoglycan and damage bacterial cell walls. So how an antibiotic works, whenever we are drinking, if you are having any particular disease or something, we will intake antibiotics, right? The antibiotics, actually in previous class, we have already told that against virus, we cannot use antibiotics. The antibiotics are used against bacteria. So what the antibiotics are doing, the antibiotics are targeting the peptidoglycan layer. So we have already told that in bacterial cell wall, peptidoglycan is there. So this peptidoglycan is targeted by whom? The antibiotics, the chemical substances present in the antibiotics. Okay. So there is a method known as gram staining. On the basis of gram staining, we can classify the bacteria into two, gram-positive bacteria and gram-negative bacteria, right? What is the difference, when we are staining by using this particular chemical substance, crystal violet, and we are staining gram-negative bacteria, after staining, there will not be any color in gram-negative bacteria. Whereas in the case of gram-positive bacteria, as it is having more peptidoglycan layer, the stain, that is crystal violet stain, will be trapped in the peptidoglycan layer and it will give that violet appearance. Okay. That red-colored appearance will be present there. Okay. So this is the gram-positive bacteria. We look into the case of gram-positive bacteria. This is a peptidoglycan layer. The bacteria cell wall membrane is here, protein is here, and peptidoglycan layer is there. In the case of gram-positive bacteria, if you are looking, peptidoglycan traps the crystal violet. Crystal violet will be trapped in the peptidoglycan layer. Whereas in the case of gram-negative bacteria, what will happen, in the case of gram-negative bacteria, peptidoglycan layer, that is present in between. There is a plasma membrane, and after that, there is a small layer of peptidoglycan and an outer membrane is there. That outer membrane contains lipopolysaccharide. Whereas in the case of gram-positive bacteria, plasma membrane is there, and after that, peptidoglycan layer is only there. So antibiotics are targeting this peptidoglycan layer, right? Whereas in the case of gram-negative bacteria, antibiotics cannot reach into the peptidoglycan layer. So in the case of gram-negative bacteria, that's why the gram-negative bacteria are antibiotic resistant. Whereas the gram-positive bacteria are prone to antibiotics. Antibiotics can kill the gram-positive bacteria because the antibiotics, the chemical which is coming from the antibiotics, can directly act on this peptidoglycan layer. So the difference in between gram-positive and gram-negative bacteria with respect to the staining process, in gram-negative bacteria, crystal violet is easily rinsed away. You can easily rinse the crystal violet and it will be revealing the red ring. It is the toxic bacteria. This is known as the toxic bacteria. This in gram-negative bacteria, it will be revealing a particular red dye. Okay, whereas in the case of gram-positive bacteria, the peptidoglycan will trap the crystal violet. Okay, so it is the toxic bacteria. Why it is known as the toxic bacteria? Because it is having another layer. Less peptidoglycan layer is present there. Antibiotics cannot act on gram-negative bacteria. So the gram-negative bacteria is less prone to antibiotic, and that's why they are toxic bacteria. Okay. A polysaccharide or protein layer called a capsule covers many prokaryotes. In many prokaryotes, if you are looking, you can see a polysaccharide or a protein layer will be covered over this, that is known as capsule. In the case of viruses also, we have told that there were capsule. Some prokaryotes have fimbriae, also called attachment pili. That is another term. Some prokaryotes will be having a particular structure that is known as fimbriae, or you can call it as attachment pili, which allow them to stick to their substrate or other individuals in a colony. When it is forming a colony, the pili which is coming from the attachment pili, which is coming from that attachment pili is known as fimbriae. Fimbriae, which is present on the cell membrane of the prokaryotes, they will help the bacteria to form colonies. They will attach together, or even they will attach to the surface also. Sexipili are longer than fimbriae and allow the prokaryotes to exchange DNA by conjugation. What is conjugation that we are going to discuss about? Sexipili is another type of pili that is not like fimbriae. It is longer than fimbriae, and they allow prokaryotes to exchange DNA by conjugation. By means of conjugation, the bacteria will exchange their genetic material, that we are discussing in the coming slides. Okay. So if you are looking bacteria fimbriae, allow bacteria to adhere to each other and a substratum, the structure that is coming from the bacteria, that you can call it as the fimbriae. These all are fimbriae. Look, most mortal bacteria propel themselves by flagella. Flagella is a locomotory structure in the case of bacteria that are structurally and functionally different from eukaryotic flagella. It is not like eukaryotic flagella, in the case of if you are seeing human spermatozoa is also moving with the help of flagella. But the flagella that is present in a spermatozoa, that is different from the prokaryotic flagella. In heterogeneous environment, many bacteria exhibit taxis. What is taxis? The ability to move towards or away from certain stimuli. So they can do the taxis. You might be heard about chemotaxis. Chemotaxis, phototaxis, all these are movement that is shown by organisms with respect to the environmental cues. So the bacteria are also having the taxis property. They can move towards or away from certain stimuli. Prokaryotic cells lack complex compartmentalization. Some prokaryotes do not have specialized membranes that perform metabolic functions. So look here in the case of bacterial flagellum, that is structurally different from eukaryotic flagellum. So here it is a plasma membrane and here it is a cell wall. So there is a basal apparatus which is attached here. From the basal apparatus, you can see one hook and one filament is there. This is a structure of a flagellum. Okay. Whereas in the case of spermatozoa, if I am drawing a spermatozoa figure here, okay, listen. Suppose if I am drawing the figure of a spermatozoa, it will be in this manner. Okay. Suppose it is the sperm. Okay, it is a sperm head. This is sperm head and this is sperm tail. So in the sperm tail, you can see proximal and distal centriole. From the distal centriole, microtubules will be coming. The microtubules, this is a microtubule. This is known as microtubule. The microtubules which is coming from the distal centriole, that together forms the tail. That together forms the sperm tail, that forms the sperm tail. Whereas in the case, this is not similar to the flagella of eukaryotic organism. It is simply flagellum and it is simply a filament is present there. That is clear. Okay. So moving to the next. Bacteria often have specialized internal membranes. If you are looking at an aerobic prokaryote, you can see what? You can see a respiratory membrane that is present inside the bacterial body. So specialized internal membranes will be also present in bacteria. In the case of photosynthetic prokaryote, photosynthetic prokaryote, that is in the sense which can produce, which can trap the sunlight and they can synthesize their own food particles and they can release oxygen. Such organisms will be having thylakoid membranes present inside that organism's body. Okay. If you're looking into prokaryotic genome, if you're looking into prokaryotic genome has less DNA than the eukaryotic genome. In eukaryotic genome and the prokaryotic genome, the major difference is that they are having less DNA. Most of the genome consists of a circular chromosome that you might have studied in your molecular biology topics. They are having circular chromosome. Chromosome is circular. That's why the replication in the case of prokaryote is known as which type of replication? That was known as in prokaryotes, the replication was
theta.
theta, because it is having circular chromosome. Some species of bacteria also have smaller rings of DNA called plasmids. In addition to the main DNA, they will be having a smaller rings of DNA. Such things are known as plasmids. The typical prokaryotic genome is a ring of DNA that is not surrounded by a membrane and that is located in nucleoid region. If you are looking in the case of a prokaryote, if you are looking in the case of the major difference between a prokaryote and eukaryote, suppose in the case of, this is a eukaryote will be having a central region. Inside the central region only, you can see, inside the nucleus only, you can see the DNA. Whereas in the case of prokaryotes, if you are looking in the case of prokaryotes, in the case of prokaryotes, if you are saying the circular DNA will be present in the central region, this central region is known as nucleoid. There will not be any nucleus. The circular DNA will be present in this particular portion only. There is the DNA is negatively charged and there is some positively charged basic proteins will be there. With the basic protein, they will be trapped in the central region. Okay, that is a case of eukaryotic genome organization. All such things you will study in your molecular biology portions. Okay, ready. If you are moving further, so here it is about the chromosome. If you are looking, you can looking in the case of a prokaryotic organism at the central region, you can see the circular chromosome. In addition to the circular chromosome, you can see the single circles here. All these single circles, they are known as plasmids. This one is a main structure, that is chromosome. And in addition, you can see plasmids also in the case of prokaryotes. Prokaryotes reproduce asexually quickly by binary fission and can clone divide every 1 to 3 hours. Binary fission, that you know very well. A single cell is getting divided into two with the help of fission, with the help of a division that is occurring at the center of the body. Many prokaryotes form metabolically inactive endospores, which can remain dormant and viable in harsh conditions for centuries. In the case of prokaryotes, if the environmental condition is not suitable for their growth, they will convert their body into, or they will produce a particular structure known as endospore, and the endospore will be remain in inactive form, that is dormant form, for centuries. And after centuries, after years, what will happen when the suitable climatic condition is coming, again from the endospore germinate to produce viable bacteria. Prokaryotes can evolve rapidly because of their short generation time, In the case of prokaryotes, prokaryotes will be having lesser evolution time. In the case of a bacterial colony, within minutes, the bacteria will be getting developed and evolving into another species. We have seen in the case of corona and all, they were producing more and more varieties, Delta variant, Omicron variant. In that manner, different variants were producing. If you have to see the evolution in the case of a eukaryotic organism like fish or pole, that will take centuries or that will take millions of years. Whereas in the case of this organism, whether the evolution depends upon the lifespan of an organism. Okay, if the lifespan is in seconds, within millisecond evolution will take place. Million seconds in the sense that will be taking only days, right? If the lifespan is in years, for example, in human beings, our lifespan is in years. 50 to 60 years is our lifespan. So as our lifespan is in years, what will happen? As our lifespan is in years, for evolution also, it will take millions of years. Okay. So this is a structure endospore, that adaptation to survive dormant harsh environment. This is a structure of an endospore. So this endospore can remain dormant for centuries. Okay. So if you are looking, bacteria evolved rapidly in response to environmental condition. One experimental condition is there, and one old tube discharged after the transfer, and a new tube is taken here. So the fitness relative to ancestor and generation, that is showing in this figure. Okay, we can discuss about this diagram later after studying about the things. Okay, come here. Prokaryotes have considerable genetic variation. 3 factors contribute to the genetic variation: rapid reproduction, mutation, and genetic combination. Genetic recombination. What is the prokaryotes are having high reproductive rate. What is the point? Prokaryotes have considerable genetic variation. Variation will occur very fastly. In the case of prokaryotic organism, variation will occur very fastly. That means they will evolve very faster. That I have already told. They will evolve very faster. Within a short span of time, a bacteria that you might have been seen in the case of some advertisement, it will kill 199.9% germs. In that manner, we have seen some advertisements. So what happened to that 0.1% germs? They are having They are having resistance. They are having resistance in the sense, after that, when after that, who will be reproducing from that area? We are using that chemical to clean our floor. 99.9% of germs were died, and only 0.1% is remaining there. Then who will produce offsprings? That 0.1% will be producing offsprings. So after that, when we are applying the same chemical, what will happen? No one will die, isn't it? No one will die from that region, because they are the mutant varieties, or they are evolution has occurred to that particular group of organism. So why the prokaryotes are having considerable genetic variation, or why they are evolving very faster? That is because of rapid reproduction, and that is because of mutation. What is mutation? Mutation is a sudden heritable change in the genetic material. Mutation can be caused by many factors. X-ray, gamma ray, in physical agents, and even chemical mutagens are there. Physical mutagens are there. All those chemical substances can cause mutation. So due to mutation also, the organisms evolve, or organism will or variation will occur in their genetic material. And what is genetic recombination? Genetic recombination is a reshuffling, isn't it? Genetic recombination that you have already studied, crossing over and all. Due to the genetic recombination also, what will happen? Reshuffling, isn't it? What is crossing over? Crossing over is in between, when we are looking in the case of chromosomes, they are exchanging their chromosomal materials, right? They will be exchanging their materials. And after that, if one organism is showing what is parental properties are and recombinant properties are. Parental property means if the offsprings are showing the character of parents only, or if they are showing the mixing up of population, mixing up of character, that we can tell it as recombinants. They can be called as recombinants. So due to the genetic recombination also, what will happen? The diversity will occur, new organism will arise. Prokaryote reproduce by binary fission. Already told that this and offspring cells are generally identical. As it is binary fission, the kids will be identical because it is getting produced from a single cell. Mutation rates during binary fissions are low, but because of rapid reproduction, mutation can accumulate rapidly in a population. What is happening in their case? They are showing rapid reproduction. Rapid reproduction means higher number of offsprings are getting produced in a short span of time. So there can be an error in the molecular machinery during replication and all. During replication, you know that during replication, a DNA has to be replicated. DNA has to be multiplied. You have to multiply the DNA. So for that, certain enzymes are there, and after that, there is a proofreading machinery also. Because within a short span of time, if you are looking in the case of prokaryotes and all, the time required for dividing their nucleus, that is less than 20 minutes. So within that 20 minutes, all the base pairs have to be multiplied. So there is a chance for error. So repair mechanism is also there in the case of prokaryote. In the case of this replication, certain enzymes are there which are needed for proofreading. And if there is any mismatch, they will do replication. They will do repair. Even though all these machinery, all these mechanisms are there present inside organism, there may be a chance for what? There may be a chance for certain errors. Such errors will cause mutation. So mutation rates during binary fission is very low. Binary fission means a single cell is getting divided into two. So normally in the case of the binary fission, the mutation rate is very low. But in the case of these prokaryotes, as there is rapid reproduction rate is very high, Because of that, mutation can accumulate rapidly in a population. High diversity from mutation allows for rapid evolution. That's why in the case of prokaryotic organism, you can see rapid evolution. Okay. Prokaryotic DNA from different individuals can be brought together by transformation, transduction, and conjugation. So that we'll discuss in coming slides. A prokaryotic DNA, a prokaryotic cell can take up and incorporate foreign DNA from the surrounding environment in a process called transformation. Transformation. Griffith was a scientist behind the transforming principle. Transform principle in earlier days, before previous days, the scientists were knowing that there is a particular chemical substance in all the cells which is responsible for determining the character, but they were not aware about that chemical is DNA. They were having several assumptions that chemical can be DNA, or that chemical can be protein, or even that chemical can be RNA. In that manner, several opinions were there among scientists regarding that particular genetic material, which is the actual genetic material. Anyhow, Frederick Griffith's transforming principle, you might be remembering his experiment by using in the case of, they were using the pneumonia organisms, Streptococcus pneumoniae, S strain and R strain. You might have you should remember all those things. S strain and R strain, smooth bacteria were there and rough bacteria were there. What was the property of S? S were having smooth polysaccharide coat, and R bacteria were not having the smooth polysaccharide coat. Anyhow, meanwhile, by using the experiment in rats, I am not explaining that experiment. Meanwhile, he was doing the experiment in rat. He could find that the R bacteria is getting converted into S form. So the reason why it was happened, from the S bacteria was killed. You might remember all these experiments. That maybe from your +2 standard itself, you are studying that particular experiment. So in R bacteria and S bacteria were there, when he was mixing R and he tickled S and R, the R was converted into S. What was the result of that? What was the reason for that? From heat tickled S, the DNA was reaching into the body of live R, and the live R was becoming as live S. So, one second. Hello? Arana? Tazeen. When he will go? Really? So, yes, we are coming, but can we come 8:30? Actually, we are a little bit away. நீங்க 8:30 சொன்னதுனால we are not near. கொஞ்சம் டிஸ்டன்ஸ்ல தான். இப்போ வந்தாலும் ஒரு 8:30 ஆகும் அங்கே வரதுக்கு. Okay, thank you. Sure, we will come. Okay, sorry for that. My kid is not doing well, so I have to go to hospital. So from hospital only they made a call. Okay. Anyhow, after this, by 8 or 8:15, we will wind up today's session, okay? Because I have a medical emergency. Anyhow, you might be remembering that experiment, right? If you are looking, there were two type of bacteria were there. This type of bacteria, one bacteria was known as R bacteria. Another one was known as S bacteria. R bacteria and S bacteria was there. S bacteria was having a polysaccharide coat. S bacteria was having a polysaccharide coat. Okay. The reason why the S bacteria is having a polysaccharide coat, that is because the S bacteria contains the DNA. The DNA contains a gene. That gene will be responsible for producing the polysaccharide coat. When he made a mixture, he killed the S and made a mixture of R plus S. What happened? The R was converted into S. R was able to develop the polysaccharide coat, and R was developed into S. The reason why, the DNA was transferred into the body of R, and the R was converted into S. Okay, that is only told here. A prokaryotic cell can take up and incorporate foreign DNA from its surrounding environment in a process called a transformation. That process is called as transformation. Griffith called that chemical as transforming principle. And after that, Oswald, Overy, McCarthy, MacLeod, these were the scientists they were proved that that material was DNA. Okay, I'm not going into those things. That is in molecular biology. Okay. Transduction is a movement of genes between bacteria by bacteriophages. That you have remembered. We have already told this thing in yesterday's class. Transduction is a movement of genes between bacteria by bacteriophage. Bacteriophage is a virus that infect bacteria. Okay. You might be remembering prophages and all. Okay. If you're looking in the case of transduction, you can see here, a bacteriophage is entering, bacteriophage is transferring its own genetic material. And after entering the genetic material, what will happen? They will divide or they will destroy. What is the cycle? What may be the cycle? Tell me about this cycle, whether it is lytic or lysogenic. By looking into the second diagram, this is. Yes, come on. This is lytic cycle, no doubt at all. Okay. So look here, what is happening? What is happening here? The bacterial DNA is getting degraded, and the viral particle that is getting multiplied. And here, if you are looking in the case, what is happening. If you are looking in the case, that is getting trapped. For example, one particular portion that is represented as A plus, and another portion is represented as B plus. That region, if you are looking, that region that is getting incorporated into the viral body. So that is a transduction, movement of genes between bacteria by bacteriophages. That is a transduction procedure that can be called as transduction. Okay. So when this A plus, when it is coming into another bacteria, when it is coming into another bacteria, suppose that A plus is reached into this bacteriophage. When this bacteriophage is affecting another bacteria, that bacteria is having A minus and B minus. That is A negative and B negative. That is the genes present here. Okay. So what will happen here? If you are looking, that genetic material from the virus is coming, and in between the bacteria and the bacterial DNA and the viral DNA, a particular exchange is taking place. This is lytic cycle, and this one may be causing lysogenic cycle. So they are getting incorporated as a prophase here. They are getting incorporated as a prophase. So A plus is coming here. And what is it happened here, that A plus and it is now converted into this bacteria which was having the gene A negative and B negative, now get converted into A plus and B negative. So from this bacteria, one A plus gene that is reached into this bacteria. That process is called as transduction. Okay, that is transduction. Study the theory very well. Based on all these things, the question, how the question will be coming, that will be exactly different. The question may be like this. These are the genes present in a particular bacteria. One bacteriophage has came, and after that, what will happen in the recipient bacterial cell? In this manner, only the question will be there. They will not be mentioning anything about transduction and all. But the theory part, that should be very clear for you. What is transduction? What is transformation? All these things should be clear. Okay. Now it's about conjugation in plasmid. You know what is plasmid? You know very well. That was circular DNA, right? So conjugation is a process where genetic material is transferred between bacterial cells. That is another process, conjugation. Between two bacteria, the genetic material will be transferred. That is known as conjugation. Now, you know very well about what is a transformation. You know very well what is a transformation. Transformation means from a bacteria, transformation from a bacteria, or that may be a died bacteria, the genetic material is moving into another bacteria, or a bacteria is absorbing the genetic material from its surrounding into its body, and they are making that genetic material as its own. That is transformation. What is transduction? Transduction means that is a bacteriophage, sir. A bacteriophage is helping for the transfer of one gene from one bacteria into next. That was transduction. Whereas in the case of conjugation, that is the direct transfer of genetic material between two bacterial cells. Sex pili, that we have already told about sex pili. Sex pili allow cell to connect and pull together for DNA transfer. So a piece of DNA called the F factor is required for the production of sex pili. All the diagrams are there. We will look into all those things. The F factor can exist as a separate plasmid or as DNA within the bacterial chromosome and is transferable during conjugation. Looking to the point once again, what is conjugation? Genes are transferring from one bacteria to next. So who is helping for that? That is the duty of sex pili. That was some longer flagella, right? It was a duty of sex pili only. A piece of DNA called F factor, that is a piece of DNA that is required for the production of sex pili. Sex pili is only connecting between, that is a connection between two bacteria. And for the production of that sex pili, what we require one F factor. The F factor can exist as a separate plasmid. What is F factor then? The F factor will be present as a separate plasmid or as a DNA within the bacterial chromosome as it is transferable during conjugation. The F factor will be also transferable during conjugation. So this is a conjugation, this is one bacteria and here it is another bacteria. In between these two bacteria, the sex pili connection is there. Okay, ready. So now look into the figure very well. This is a bacterial chromosome. This is a first bacteria and this is a nearby bacteria. This one is represented as F plus. It is contained in that F factor, that is F plasmid is present here. This is known as F minus cell. F minus cell means it cannot produce what? It is not producing sex pili. So sex pili will be produced by this bacteria. So from this F plasmid, what will happen? Look here, the F plasmid, that is moving and that is producing the sex pili. The sex pili is produced here. So here this bridge can be called as the mating bridge. So through the mating bridge, what will happen? Look here very well. Through the mating bridge, the F factor is transferring from this bacteria, that will replicate and that will transfer into this bacteria. Whenever it is reached into this bacteria, both of the cells are now becoming as F plus. This one is F plus now and this one is also F plus now. It is clear, right? Okay. So this is a conjugation. This is a conjugation. This you can see as a conjugation. Now conjugation and transfer of a F plasmid. So this can be called as HFR cell. So there is F factor that is present as separate. There are two cases. In one case, in one case, the F factor will be present as a separate plasmid, or the second case, the F factor will be present inside the DNA. So in this case, that DNA is containing the DNA itself is containing that F factor. Okay, DNA is containing the F factor. Okay, ready. So this is F minus cell. It is not having any plasmid. So if you are looking in this case, if you are looking in this case from this point, the DNA through this mating bridge, the DNA along with who is coming first, that is that F factor is coming first. That F factor is coming here and F factor is reaching here through the replication process only, duplicating, and it is moving here. And slowly that is reaching here, it is reaching here. And this one is already HFR. And after that, what will happen? This will become as, this will be containing what? This will be containing the F factor. F factor is present and its own DNA is also present here. Look here, the A plus which is present in this DNA now reached into this one. It was containing A minus only, A minus, it was containing A minus, and that A plus that is was present, let it be a gene or let it be a segment of DNA or anything, that is now reached into the F negative bacterium. So this is the theory behind this, but the question will be the genes present in F plasmid. In that manner, you will be having question. For example, F plasmid contains gene number A, B, C, D, E is there. And in that manner only, so what will happen after conjugation? So in that manner only, you will be getting the question. When we are discussing the question, it will be more clear. But now you have just understand about what is conjugation and what is the two types are there, right? One type, the F factor is present as a separate plasmid, and in this case, the F factor is present inside the DNA. Okay. R plasmids are antibiotic resistance. R plasmids carry genes for antibiotic resistance. R plasmids. That is another plasmids that carry genes for antibiotic resistance. Antibiotic selects for bacteria with the genes that are resistant to the antibiotics. Antibiotic resistant strain of bacteria are becoming more common. So, what will happen? The bacteria, bacteria, if you are continuously using antibiotics against particular bacteria, that will result only the selection of resistant varieties. If you are continuously intaking a particular antibiotic, what will happen? After few months or few years, what will happen? That bacteria will get resistance against the chemical substance present in that antibiotic. Okay, that is clear. Then diverse nutritional and metabolic adaptation are involved in prokaryotes. There are phototrophs, they obtain energy from light and they can synthesize their own food. Chemotrophs are there, they obtain energy from chemicals. Autotrophs are there, they require carbon dioxide as their carbon source or their carbon source. Autotrophs are there, they are depending upon phototrophs for their energy. Whereas phototrophs and chemotrophs, they can produce their own energy. Heterotrophs requires an organic nutrients to make organic compounds. These factors can be combined to give the four major modes of nutrition: photoautotrophy, sir, chemoautotrophy, sir, photoheterotrophy, sir, and chemoheterotrophy, sir. Heterotrophic organism will be depending upon the phototrophic organism. That is clear. Autotrophic organism. So nutritional diversity, if you are looking, photoautotrophs are there. Their energy source is light, carbon source is carbon dioxide, and the type of organisms, there is cyanobacteria and algae are there, that is photoautotrophs. Even our plants are also coming under this category. Looking into chemoautotrophs, you know who is chemoautotrophs? They are sulfolobus. They are example for this organism. They are using inorganic chemicals and their carbon source is carbon dioxide. In the case of photoautotrophs, they are using light energy and chloroplexus and rhodobacter. These are our examples. Chemoautotrophs are using organic compounds as their energy source and also as their carbon source. The examples are Clostridium, protist, fungi. All these are examples for that. Okay, by heart this table. Prokaryotic metabolism varies with respect to oxygen. There is obligate aerobes. They require oxygen for cellular respiration. That is obligate aerobes. They cannot survive without the absence of oxygen. Obligate aerobes. Obligate anaerobes are there. They are poisoned by oxygen and use fermentation or anaerobic respiration. In the case of obligate anaerobes, you know, they cannot survive in the condition if there is oxygen is present. They are doing the fermentation and all. Lactobacillus, you may be remembering about the organisms like Saccharomyces cerevisiae, all those organisms. Facultative anaerobes are there. They can survive with or without oxygen. That is facultative anaerobes. Okay. In nitrogen fixation, what is nitrogen fixation? Atmospheric nitrogen. Nitrogen is present in atmosphere in large amount, but the soil is, the plants cannot utilize that atmospheric nitrogen directly. So the nitrogen should be made available to the soil through a process known as nitrogen fixation. Nitrogen has to be converted in the form of nitrate. That is nitrogen fixation. Some prokaryotes convert atmospheric nitrogen to ammonia, NH3. Metabolic cooperation between prokaryotes allow them to use environmental resources they could not use as individual cells. So there is a metabolic cooperation between prokaryotes allow them to use environmental resources they could not use as individual cells. In the Cyanobacterium anabaena, photosynthetic cells and nitrogen fixing cells called heterocytes exchange metabolic products. So there are even association between organisms that you can see. You might remember about the leguminous plants, you know, in the leguminous plants, in pea plants and all, their root nodules are containing a particular bacteria. That bacteria is known as Rhizobium leguminosarum. They are fixing atmospheric nitrogen. That's why in paddy fields and all, we are cultivating in summer season. After harvesting the paddy, we are doing pea cultivation. That is for making the nitrogenous bacteria, the plenty amount of nitrogen. We have to convert the atmospheric nitrogen to nitrate. That's why we are doing that particular thing. The nitrogen is very much essential for plant growth and development. Okay. Molecular systematics is leading to a phylogenetic classification of prokaryotes. So we can look into their evolutionary relationship. It allows systematics to identify major clades. The use of polymerase chain reaction PCR has allowed for more rapid sequencing of prokaryotic genome. All these are simple theory points. A handful of soil may contain 10,000 prokaryotic species. That is the number of species only. So million numbers of bacteria will be present there. Horizontal gene transfer between prokaryote obscure the root of tree of life. Okay. So if you are looking in the universal ancestor, in the domain eukarya, you can see eukaryotes. In the domain archaea, you can see Coracoids and Eukaryotes, Caranarchaeodes. In this manner, you can see. In the domain bacteria, you can see Proteobacteria, Chlamydia, Spirochetes, and Cyanobacteria, gram-positive bacteria that you can see that is coming from the universal ancestor. Look here, archaea are prokaryotes and share certain traits with the bacteria and other traits with the eukaryotes. That is archaea. At the beginning of the chapter, we have told that prokaryotes, they contain both archaea and bacteria. The archaea are prokaryotes and share certain traits with the bacteria and other traits with the eukaryotes. Some archaea live in extreme environment and are called extremophiles. Extreme halophiles, that is halophiles, they are living in salty condition. Extreme thermophiles, they are living in the hot environment. Okay. Archaea appear to be more closely related to eukarya than bacteria. The archaea, that is, appear more closely to the eukarya. If you are looking into the evolutionary, you can see eukarya will be eukarya and archaea, they are more related, and bacteria is not that much related to archaea. Okay, it is related but not up to the relation matrix shown by the eukarya and archaea that you can see from the diagram. So there is a representation of the comparison of that tree that based on these domains. If you're looking into different organism, bacteria, archaea, and eukarya, you can look into this diagram. You can look into the chart, and you can have the major difference between for your B part questions and all. You can get question from this area. In bacteria, nuclear envelope is absent, whereas in archaea it is absent, whereas in the case of eukaryotes it is present. Membrane closed organelle that is absent in bacteria, absent in archaea, and present in eukarya. In this manner, you can look into all the cases. You have studied in molecular biology that methionine is the first amino acid, right? That is a initiator amino acid. But if you are looking in the case of bacteria, it is not methionine. It is formyl methionine. In the case of archaea, that is methionine. In the case of eukarya, that is methionine. But in the case of bacteria, that is formyl methionine. Even though the formyl methionine is the first amino acid, in all the protein, the first amino acid will be methionine only. How it is, there is a particular enzyme known as deformylase. That deformylase will cut the formyl group in the case of bacteria, and after that the methionine will be present. Methionine only will be present in the case of bacteria also. Okay, if you're looking into this thing, you can see, look at the case of circular chromosome, it is present in bacteria. Archaea is also containing circular chromosome, that is plasmid, but it is not present in the case of eukarya. And in this manner, you can look into each and every point about RNA polymerase, about membrane lipid, about initiator code amino acid, and introns in genes. Introns, what is intron? In genes, you can see introns and exons, right? Introns are the intervening sequence. If you are looking in RNA contains both introns and exons. I am not calling all those things. All those things you are studying in molecular biology, you know very well. Introns means intervening genes, which are not coding for proteins, which is present in the case of bacteria. That is very rare. Very rare in the sense they are having very smaller DNA. In the DNA, almost all the nucleotide sequences are coding. They are not having enough size of DNA to accommodate all the unwanted things. Whereas in the case of eukarya, if you are looking, eukarya, it is having a large size of DNA. As it is having a large size of DNA, a number of nucleotides will be also present there. And almost all the nucleotides, someone will be coding and someone will not be coding for amino acids. Okay. What is methanogens? Methanogens live in swamps and marshes and produce methane as a waste product. Methanogens in biogas plants, and you can see this. Methanogens are strict anaerobes and are poisoned by oxygen. In recent years, genetic prospecting has revealed many new groups of archaea. Some of these may offer clues to the early evolution of life on Earth. So that all those things. Okay. Bacteria include the vast majority of prokaryotes, which most people are aware. Diverse nutritional type are scattered among the major groups of bacteria. All these things we have discussed. Rhizobium are nitrogen fixing bacteria. They form root nodules in leguminous. Mutualism. Mutualism means that is plus. Both organisms are getting benefited. Bacteria is getting benefited, and also the plant is getting benefited. The bacteria is getting a place to live, whereas the plant is getting nitrogen for their photosynthesis and fixed atmospheric nitrogen. There is rhizobium. Agarobacterium produces tumors in plants and is used for genetic engineering. E. coli residue resides in the intestine of many mammals and is not normally pathogenic. In our intestine and all, we can see E. coli. Some common bacteria. Chlamydia are parasitic bacteria that live within animals. Chlamydia trachomatis causes blindness and non-gonococcal urethritis by sexual transmission. Spirochetes are helical heterotrophs. Some such as Treponema pallidum, which causes syphilis, sexually transmitted diseases, and Borrelia burgdorferi, which causes Lyme disease, are parasites. So major groups of bacteria, chlamydia is here, spirochetes are here, cyanobacteria is here, gram-positive bacteria are here. Look into the diagrams and all when I am sharing the PPT to you. Okay. Cyanobacteria are phototrophs that generate oxygen. Plant chloroplasts likely evolved from cyanobacteria by endosymbiosis. Look into the point very well. Cyanobacteria, they are photoautotrophs that generate oxygen. So the cyanobacteria are bacteria. The cyanobacteria, if I am representing by using a diagram here, cyanobacteria, that is, okay, cyanobacteria, I am representing the figure of Cyanobacteria is here. Okay. A cyanobacteria. What is the property of this organism? Cyanobacteria, they are photoautotrophs. They can generate oxygen. Plant chloroplast likely evolved from cyanobacteria by endosymbiosis. This is a plant, okay? This is a plant. This is a plant cell. The plant cell is having a property to produce what? The plant cell is having the property to synthesize food by photosynthesis. How it is happened, you know, that is by the process of the endosymbiosis of this organism, that is this bacteria, that is reached into this plant's body. In the case of evolution, this bacteria was reached into this organism's body, this plant's body. Then only the plant had got the ability to produce the oxygen by doing photosynthesis. Whenever the plant cell divides, you know, the bacteria also divides. That is a chloroplast. That's why the chloroplast is containing its own DNA, chloroplast DNA. Likewise, in the case of mitochondria also, mitochondria is also reached into organism's body by the process of endosymbiosis only. The chloroplast and mitochondria in the point of evolution, you know, they were actually microorganisms. They were reached into the organism's body and get incorporated there. Okay. Next, gram-positive bacteria. Gram-positive bacteria includes actinomyces, which can decompose the soil. Bacillus anthracis is that cause anthrax, Clostridium botulism, food poisoning, right? Staphylococcus and Streptococcus, which are pathogenic. They are causing different diseases to human beings. Mycoplasma are the smallest known cells. All those things are clear. Prokaryotes play a major role in the recycling of chemical elements between the living and non-living component of ecosystem. You know very well about the sewage treatment. In sewage treatment, you know, the microbes are only treating the sewage. Till now, no human-made technology can detoxify all the sewage and convert them into the water which can be released into natural water bodies. Chemoheterotrophic prokaryote function as decomposers, that is saprophytes, breaking down corpse, dead vegetation and waste products. Nitrogen fixing prokaryotes are usable nitrogen to the environment. Prokaryotes can increase the availability of nitrogen, phosphorus and potassium for plant growth. Prokaryotes can also immobilize or decrease availability of nutrients. So here it is about the now the time is already you have to then I can complete this. Symbiosis is an ecological relationship in which two different species live in close contact. The larger host and smaller symbiont. When two organisms are living together, we can call it as symbiosis. In mutualism, that is plus. Both organisms are getting benefited. In the case of pea plant and Rhizobium leguminosarum, we are told that. In commensalism, that is plus and zero. One organism getting benefit and other organism, while neither harming or nor helping the other. Other organism is not getting any benefit and it is not harming that organism also. Whereas in the case of parasitism, you know, an organism called a parasite harm, but it does not kill the host. Parasite that cause disease are called pathogens. In parasitism, you know, one organism is getting benefit and other organism, you know, some problem is occurring in other organism. It will not kill the host, okay? Pathogen bacteria causing Lyme disease are transmitted by deer ticks. Pathogenic prokaryotes typically causes disease by releasing exotoxins or endotoxin. What is exo and endotoxin? The exotoxin causes disease even if the prokaryote that produce them are not present. Exotoxins are cause disease even if the prokaryote that producing them are not present. So exotoxin itself can produce disease. Whereas endotoxin are released only when the bacteria die and the cell wall breaks down. That is endotoxin. So endotoxin will be produced in an organism's body in our body. Whenever that bacteria die, then only the endotoxin will be released out. Many pathogenic bacteria are potential weapons for bioterrorism. Now, war is occurring, right? We are not going into the topic and all. There is a particular term means biowar. Okay. If you are going towards that, it will take more time. Experiments using prokaryotes have led to important advances in DNA technology. Prokaryotes are the principal agents in bioremediation. What is bioremediation? The use of organisms to remove pollutants from the environment. Do you hear about superbugs? Superbugs are Govind Khorana, that was an Indian scientist, and you might be remembering, for removing oil from oceans. We are using superbugs. Okay, leave all those things. Some other uses of prokaryotes are recovery of metal from ores, synthesis of vitamins, production of antibiotics, hormones, and other products. We are using prokaryotes for all these things. So it says general structure of a bacteria. We have already told there is a circular chromosome. In addition, plasmids will be there, cell wall will be there, and even fimbriae is there, sex pili is there, flagella for movement. All these structures will be present there. And in certain organisms, internal membrane will be also present there. Look, in the case of nitrogen fixing bacteria and leguminous plants, what is their benefit? It is represent in the absence of rhizobium after 12 weeks of growth, a plant that have mass is about 0.1 gram only. Whereas in the case of this is present, if it is present, what will happen? If the rhizobium is present, it will fix atmospheric nitrogen and plant will get more amount of nitrogen. If it is getting more amount of nitrogen, that can produce more nitrogen bases and even that can be used for replication and protein synthesis. For everything, they can use that nitrogen. Okay. So in today's topic, now it is very clear that how to distinguish between the cell walls of gram-positive and gram-negative bacteria. That is clear. State the function of the following features. What is capsule? You know very well. What is fimbriae? What is sex pili? What is nucleoid? What is plasmid? And what is endospore? You have to answer me. Explain how R plasmid confer antibiotic resistance on bacteria. That you know very well. That is R plasmid. That is another type of bacteria, right? R plasmid. Another type of plasmid is R plasmid. F plasmid you know very well, and R plasmid is also very well known now. Explain the importance or uses of prokaryotes. That is simple only. Look into all the three questions and give me the answers. And we are winding up today's session here because I have to go to hospital. Let me close the record now. Stop the recording now. Stop recording.